half you guys that have sent an email. I think you're about 20 so far, 20, 22. If you haven't done so far, you should do it right now. You have like a couple minutes before we get started. So shoot me a quick email. That way I know your preferred email address. And what that's going to do is going to give me the email that I'm going to use to email you guys the links for Google Drive, which I'll address in just a bit, and also an invitation to Canvas. And as I mentioned on Tuesday, Canvas isn't mandatory, and it's not supported here by West LA for my class, specifically because this is an in-person class. I'm just using Canvas. Canvas is free for teachers to use. And it gives me the ability to kind of just centralize all that information into one and allow you to see your grades real time. Otherwise, I would just leave you guys to calculate your own grades. And I know people don't really do that until towards, when it really counts, especially towards finals. So I want you guys to be aware of your progress as we go through. I'll update real time and you guys will be able to see how you can do. One of the advantages that I've seen, which I've been able to implement in previous years, is that towards the finals, you'll be able to go in and see what your projected grade would be on your final grade. So if you say, I want like, I don't know, at least an A- on the final, you could see what your projected grade in the class would be. So it's no surprises. So for those that need to get that bump in your letter grade, you know how well you have to do. That way you can kind of gauge your efforts and know exactly how well you need to study in order to get the grade you want. And then also I've seen the converse where people have like, let's say like a 95 in the class. What do they need to do in order to keep that A in class? And you see them get like a 75 and they're like, all right, I got a 75, you still get an A in the class. That way you can at least see with some accuracy how well you need to do, all right? Now, Google Drive, who's had access? Who's been able to get access to Google Drive with the links that I sent out? And is it for 2017 fall? Do you guys see that, or is it 2017 spring?
I don't know whatever you send, I see it.
There's two. The first one you send out with the email, requesting the email address, that one gives you both spring of 2017 and fall.
Yeah, for some reason there's like a bug on Google Drive. I have to think, what I'm going to do is just create a new folder and I'm going to send out a link because for some reason it's trying to send out spring 2017.
The second one, it's asking us to request access.
Yeah. So I'll send something out because it works. I've tried it on my computer. I've tried it on one computer. It gives me 2017 fall. I tried on my computer and it's trying to link me to 2017 spring, which is not correct. But anyway, you look at it, you guys don't have access to spring material, do you? Okay, cool. So let me send out the links. Once I compile everybody, I'll make sure I get that out to you guys. So with that being said, you guys don't have the PDF, right? For today, at least the the large PDFs. I'll make sure that if you guys can send me your emails at least by Friday, I'll get that whole list. I'll blast you guys with a new link. And what you're going to see on there by this weekend, you should be able to see this week's lectures. You'll see next week's lectures. You'll see the study guide for the first 2 weeks. And what else? You'll see the syllabus on there. So if you ever need access to that stuff, just bookmark those links and you'll be seeing stuff just getting dropped in there. What's also useful if you have a Gmail account is you get notifications when things get dropped in. So if you're wondering, when's Marlon going to upload lecture 12? You'll get a notification at some point that says lecture 12 now available. It's kind of the reason why I'm putting things on Google Drive so that you guys get notifications on them. Sound good? All right. We've done our rosters for today. Again, we're only doing it this week and next week. After that, I'm not requiring any attendance of any kind. So be here on the days things are due, be here for the quizzes, be here for the tests, and then you should be good. All right? Any questions before I start? All right, so what are we doing today? Just as a recap of what we talked about last week, we talked about all the different levels. We looked at the organism, we looked at the organ system, down to the organ. We've looked at the tissue level, down to the cell level, and what we're going to be doing today is we're going to be looking at the atomic level, which we're not going to spend too much time on, and we're going to spend time on the molecular level. So we're going to be going over some of the main molecules that we're going to be seeing in physiology. As we go through our lectures, you'll be able to see the references that we're going to be making. So this stuff is going to build on top of what we're going, this is going to be the foundation of the stuff that we're going to be learning when we look at some of the molecules. This is going to serve as utmost importance because we're going to be learning about things like lipids and how fats interact with the cell that you're going to learn next week. You're going to learn about how water-soluble molecules interact with a cell. Again, something we're going to learn next week. So it's prudent for you guys to understand the makeup of these molecules and the properties of these molecules before we get into physiology. You've seen this stuff before. This is all chemistry. In fact, this is biochemistry. So at some point in your academic careers, you've taken biochem. But I'm not going to delve in as deep as what you guys have learned. I'm really going to cover the basics. And again, some of the stuff you're not going to be seeing after quiz 1, but I will point out the things that will be important for you guys as we go into our later lectures. So our topics for the day. We're going to look at our atoms. We're going to look at when we put the atoms together. We're going to be looking at our molecules. And we'll be looking at the bonds that hold these molecules together. And then when we look at our molecules, we'll be taking a look at 2 types of molecules, inorganic molecules and organic molecules. And again, I'll talk more about the differences between those. And the main types of macromolecules we'll be talking about that are organic molecules, we'll be looking at DNA, protein, carbohydrates, and fats. And we're going to visit all of those today so that you understand the structure of those. And what we'll be doing next week, we'll be talking specifically about the cell and how the cell is made up with these types of molecules. And at least they'll give you the cellular biology portion. Next Thursday, we talk about the functions in the cells, mainly cellular respiration. How do we generate energy? And again, that will be the foundation before we start to kick into the more complex cells, the cell differentiation, the term that I talked about on Tuesday that refers to the specialization of cells. So now that you kind of have a foundation of what we'll talk about, let's get into it. And before I do that, of course, real quick, let me just turn something off. And I'm back in. So atomic level organization. So the structures of your atoms. You've seen, again, you've seen this before. It's just a revisit. But we're going to be looking at the contents of the atoms, which we know there's 3 main components that are typically discussed in at least an introductory chemistry course. Protons, electrons, and neutrons. These are the 3 main components. I'll talk about those on the next slide. But specifically, when we look at the elements, we know that there's 92 naturally occurring elements on Earth. That's present specifically on Earth. You might think, well, I've seen a periodical table, and it definitely looks like there's more than 92 natural occurring elements. And something we'll take a look at here, a good section of these are man-made. In fact, there are more that are being produced. Things up over here I can't even really pronounce. But these are manufactured in a laboratory. It's stable for a little bit amount of time, but it doesn't exist naturally here on Earth. So if you were to look at the major elements, remember what I mentioned, the ones that we talked about on Tuesday. There were 6 main elements that are found in the human body that comprise 98% of all elements found. And if you remember, that was PCHONs. That was the acronym I had you use. But if you were going to look at 95%, and I know I'm just kind of playing around with the percentages here, really the major elements would be carbon, oxygen, hydrogen, and nitrogen. And when we look at our complex molecules later on today, I bet you that most of that will be this, CHON. The other elements, when we look at the other 5%, some of them will include the lesser elements. Things like calcium, phosphorus, potassium, sodium, chlorine, magnesium, iron. Iron you would have seen in your red blood cells, specifically a protein called hemoglobin that we'll talk about later on that will hold on to iron. These are also required to be found in our human body, but in lesser percentages. And then trace elements. Some trace elements you're going to be able to see. We don't talk too much about them, but we will talk about an element called iodine, something that's required, something that you've seen as iodized salt. It's our intake that we'll talk about. Makes a profound influence when we look at our endocrine lecture when we talk about thyroid diseases. All right, so with that, that is our atomic level of organization. If we take a look at, you'll see that the protons and neutrons are clustered around the center, and then we have electrons that are floating around in space around the nucleus of that atom. So then, that again, you've seen before. If you were to weigh these things out, you would see that protons and neutrons weigh about 1 Dalton, otherwise known as 1 atomic mass unit. The electron has actually a negligible weight. It's about like 1 out of 2,000 of a Dalton. So if you were to look at an element, you would see most of that is clustered. Most of the mass of that atom is clustered around the center. And then you've got all these negligible weight electrons that are literally just floating around space, just circulating around. And if you look at the protons have a positive charge, and the electrons have a negative charge. And again, they circle around in these orbitals around the center of the atom. So the two numbers you'll see on here, I'm never going to ask you guys about these, so don't worry about these. But atomic number is the number of protons in an atom. And the number of protons governs what type of element it is. Okay? If you were to look at the mass number is the sum of protons and neutrons. And you might think, well, mass number, mass doesn't relate to weight. Well, we know that protons and neutrons are about 1 Dalton each. So if you count the number of protons and neutrons, that tells you exactly what your mass number is. And you'll be able to see this in our periodical table of elements in just a bit. Actually, you can even see it here. So if we look at the atomic structure, we're going to look at CHON, and this is hydrogen, carbon, nitrogen, and oxygen. These are the 4 main ones we're going to talk about. We'll talk about sodium and chlorine, maybe not so much potassium and iodine, but at least the idea I want to get across is, again, you'll see the protons in the center, neutrons in the center, and you'll see the electron floating around. One of the key things I want to point out, again from basic chemistry, electrons float around the center of that atom, but they fall around. They travel around these orbital shells. And again, I'm not going to get into too much detail about it, but the general rule is that the first shell, and you'll see these shells that are surrounding each of those proton-neutron cores. The first shell holds about 2 electrons. The second shell, and every shell thereafter, third to fourth, etc., will carry about 8 electrons each. And I know there's other orbital shells, so if you're a chemistry guru here, don't knock me on my chemistry. I'm here to teach you guys physiology. But the main idea behind it is, if you take a look at, let me go into carbon. You'll see carbon has 6 protons. That's just a fact. If you were to count the number of protons in carbon, it's always 6. If you look at the number of neutrons, in this case, we have 6 neutrons. So 6 plus 6, 6 Daltons plus 6 Daltons, for that of proteins and neutrons, give us a mass number of 12. Okay, I'll talk about this 13 in just a little bit. If we take a look at the electrons, you'll see that we have 6 electrons floating around the shell. So knowing that we have 2 electrons in the first shell, that means if there's 6 electrons, that should mean there should be how many in the outer shell? 2 electrons on the first, 4 on the outer shell. Okay? So I'll talk about that in a little bit when we talk about our carbon atom. Let's give you another example. Let's take a look at sodium. Okay? Sodium has 11 protons. Therefore, sodium should also have electron. If sodium's neutral, again, the topic I'll talk about in a bit, if sodium is neutral, it should have 11 electrons. So given that there's 2 electrons in the first shell, 8 electrons in the second shell, how many are there in the third shell? 1. Okay, so keep that in mind. We have 1 electron in the outer shell all by itself. Take a look at chlorine. Chlorine, if you were to look at 17 protons, you don't really need to do the math. You could really just look at the outer shell. It's already done for you. You can count 7 electrons. Okay? So all it needs is 1 electron on that outer shell, and then it would complete. So now that we have that in place, we know electrons are floating around. We have this general rule here. Let's take a look. Before I move on to the next slide, something I want to bring up. No, scratch that. We'll move on. The next topic we'll talk about are isotopes. And isotopes, basically, the main definition of isotopes is that atoms of an element have a different number of neutrons. And let me go back to the prior slide, where I pointed out that the mass number for carbon is either 12 or 13. So you're thinking, why is there 13? If the number of protons will never change, right? Because the number of protons in carbon is always going to be 6. If it was 7 protons, well, you wouldn't have carbon anymore, you would have nitrogen. So in carbon, if we have 6 protons, we can see that maybe there are isotopes out there that have a different number of neutrons. And in this case, if there were 13, the mass number was 13, that would mean if there's 6 protons, there's 7 neutrons. And the excess neutrons happen to actually give off a little bit of energy. It's a little bit of radioactivity that can actually be measured. So when we look at the radioactivity of that particular element, and let me just draw out 13C, the way you would see that notation for a radioactive isotope would be a 13 on there, letting you know the mass number of 13. From there, you would have to look at the periodical table of elements. You would see there's 6 protons. You would know that there's 7 neutrons. So that extra neutron does give off excess gamma rays. There's a small percentage of radioactivity that's given off. And there are applications for it. Okay. So if you have, there's 13C and then there's 14C. This means there's 2 excess neutrons, which actually gives off more radioactivity. And over time, if you had a whole cluster of these, 13C or 14C, radioactive carbon, over time it would start to die off. Because once you keep giving off energy, over time that isotope will turn into something else. What it turns into isn't really the focus of what we're going to talk about, but nonetheless, it still gives off radioactivity. So we've seen lots of applications for this. In fact, the first one I'll talk about is carbon dating. And has anybody here heard of carbon dating? Okay. So what exactly is carbon dating? Carbon dating is a way for us to be able to determine the age of a sample. Say a paleontologist finds a fossil, and they take a look at a particular fossil, and they want to find out how old is this? And they say 7 million years. Where do you think they came up with that number? You think they just made it up? No. Actually, what they do is they actually will take a sample of the bone in there, and they know that at any given time, we have a certain percentage of 13 carbon. And I don't remember exactly what that percentage is, but it should be something, let's say, 0.02% naturally occurring. That means if I go and take a look at the carbon, if I were able somehow to grab all the carbon from your body and measure it out, I would know that 0.02% of that would be this radioactive isotope. Okay? Now, if you take that old sample and I measure and I find out that the amount of 13C in there is 0.0005%, I know I've lost a certain amount of 13 carbon. And that's because over time, that radioactive isotope has decayed. Okay? So how long does it take to reduce carbon down to, let's say if I were to take like a pound of 13C, to turn into half that amount? And again, you don't really need to know this. It takes 5,730 years to turn into half of what its original amount is. That term is called the half-life of radioactivity. For a particular element, there's a certain half-life. You guys familiar with the flooding that happened in Japan after the earthquakes? They have all this radioactivity in there, and what they have to do is they have to take all that radioactivity from that nuclear power plant that exploded or whatever it did. They have to take it and they have to store it and wait many years for that plutonium to have its half-life. And it takes a long time for that stuff to decay. They have to contain it and leave it there so that it doesn't affect anybody. And over time, that stuff dissipates and it's no longer radioactive. Chernobyl happened a long time ago. There's still a lot of radioactivity out there, and it's because that stuff is still out there. So they've kind of deemed that area, don't go around because you'll pick up that radioactivity, and that radioactivity can cause things like cancer, it can cause damage to your body. So nonetheless, if they were to measure, back to our original thing that we were talking about, if they were to measure that 13C, that 13 carbon, and they found out we had a proportional amount of less, they could figure out the number of years of how old that sample is. Fairly accurately, too. All right, so we're talking about physiology. This isn't really, carbon dating isn't all that important. Maybe from an anthropology perspective, but let's talk about a clinical application. Positron emission topography. This is your PET scan. Has anybody ever had a PET scan here? Nobody? I've had a PET scan done. And what they do is you go into a scanner and they put you in a machine. And it kind of looks like an MRI, but what they do is they inject a certain contrast in you. And this contrast is called fluorodeoxyglucose, FDG. You don't need to know this. FDG is basically a glucose molecule with a tagged, and a glucose molecule is a sugar molecule that you take in, and it has a tagged radioactive isotope on there. So if I were to give this to you, and you had a little contrast, any areas in your body that's taking in that glucose will get lit up. Okay? So what you're seeing here are some images of that. They'll put that person in a scanner, they'll inject them with a contrast, and they'll watch any areas of the brain that's taking in that glucose for energy starts to get lit up. So these are areas that correspond to high metabolic activity. They do these things also with cancer too. They can give you, if they're trying to diagnose and try and find areas in the lung, for instance, that seem to be particularly lit up, like right here. Actually, I don't think this is the lung. But if they were looking at areas that were highly active, they would see, oh, this area is kind of lit up and it shouldn't really be. This warrants some investigation into that organ to see if there are some tumors in that area. So again, FDG is something that they use because it gives off a certain radioactivity, can get picked up by this machine, and they can know a lot about it. Something that I did back in college, I know I talked about this on, I think I may have talked about this on Tuesday. I worked in research, but I was also a research guinea pig. And back in college, I was an undergrad. I remember looking at the UCLA newspapers and finding like experiments that they were doing on people. And I would see some that were paying pretty well, and I'd call them up, find out, read the consent form, and it'll tell you exactly what they're doing. And I thought, they're paying $500, why not? I'll go and start doing these things. And this one thing they wanted me to do was to go in, and don't think I'm a freak just because I signed up for these. I was broke, you know, I needed some money. They had me sit into a machine. Again, they set me up, put me into the machine, into the PET scan, and actually it was an MRI machine, but that doesn't matter. They injected me with the contrast, and they wanted to look at my brain and see the amount of activity that's happening in my brain. Now, they've removed all sensory. They gave me huge earplugs. They gave me goggles so I couldn't really see anything, and they basically told me, don't think about anything. In fact, try and sleep, but don't sleep. And then they want to use that to measure the brain activity. Then they put these goggles on me, and they made me watch this video. And this video consisted of someone, and all I saw was someone picking up a coffee cup and then putting it back down, and it was empty. And I watched this video for an hour and a half. And I remember thinking, they just kept telling me, just focus on the coffee cup, think about somebody picking up the coffee cup and putting it down. And that's all I watched for about an hour and a half. And that whole time they were measuring areas in the brain that would correspond to the whole idea of picking that up. There's a spot in the brain that's specifically attuned to the action of picking something up with your arm. And they found that spot somewhere off onto the side. After I was done with that, the second part of the experiment was they'd sit me down in the chair, they put this huge helmet on, and there was a spike that came on, and they said it was going to just kind of inject a little bit right into the scalp, just a little bit, and they were going to prod that area with electricity. And I didn't do that portion. I'm like, no, you're not doing that. To this day, that happened and I know I'm kind of dating myself, that happened about 15 years ago. I can still see the images in my head. I still see that same coffee cup, and it's not like flashbacks or anything, it's just, I remember this video, right? A really interesting thing, and I bring this every time I teach it. I found the research article 3 years ago, and I read it, and they had pictures of the coffee cup, and I kind of got the shivers. I was like, ooh, this is really weird. They had like 10 people in this experiment, and it was weird knowing that I was one of those people. So, nonetheless, that's exactly what they did with me. They injected that contrast so they can find that area in the brain that's active, and that's exactly what they do in a clinical setting. All right, so here's our periodical table of elements. Again, something you would have seen in chemistry, but this tells you on here the number of protons. You can see the number of protons for carbon, you'll see 6. And then you'll see the mass number underneath. And again, that mass number is the sum of protons and neutrons, of which you can see carbon has 12.011. So if you're thinking, where's that .011? This is an average of all the carbon that's existing out there, and you take an average of it, you'll see that most of it will have 12, at least a mass number of 12. There'll be some that have a mass number of 13, and even a far less number that have 14. The 13 carbon and 14 carbon are the radioactive isotopes. And then they can use that to be able to see inside the body. They can actually tell us which areas are active. All right, so with that, I'm going to move on to the next slide. Let's start taking those elements that we looked at and start piecing them together. Let's start putting them together in molecules. Those molecules that we'll be looking at, now we've taken two elements together. We want them to hold together. And the four types of holds that you'll see on here are chemical bonds. And they'll rigidly hold, well, maybe not all rigidly, but at least hold the molecule together. The two of which we'll talk about take place within the molecule. They're called ionic bonds and covalent bonds. The third one are called hydrogen bonds, which attract each other. And again, I'll talk about these attractive forces in a little bit. And then metallic bonds, not so physiologically relevant, so I won't really talk about these. Okay. So you guys can refer to this slide because I'm going to talk about these in the next couple slides. So ionic bonds, complete transfer of electrons. Let's take a look at that in sodium and chlorine. You guys already did this for me. Going back to this prior slide, we looked at sodium and we looked at chlorine, of which we know sodium has 1 electron on the outer shell, which you guys already told me. I told you guys there's 7 electrons on the outer shell for chlorine. So let's take that example where we mentioned earlier. The thing is about atoms is that they are most stable when they have a full shell on the outer shell. So looking at sodium, you see, as I said earlier, first shell, 2 electrons. Second shell, 8 electrons. Outer shell, there's 1 electron. So in order for it to be stable, it would most likely get rid of that excess electron. So if each electron has a negative charge to it, and that sodium gives off an extra electron, is it going to have a positive charge or a negative charge? So it's going to have a positive charge. So that positive charge on here is called any charged element is going to be called an ion. And ions can be categorized. Again, not something you really need to know, but we have cation and anion. Cations are for positive ions, anions for negative elements. And a way to remember this, if you were to think, if you're a cat person, cat would be positive. Anion kind of looks like onion. Onions are negative. Unless you really like onions, then you got to think of something else. But those are the names of those types of ions. So in this case, that sodium ion would be a cation, and it has a positive 1 charge. It gave off 1 electron, so it has a positive 1 charge. So we typically would see Na+, and that plus signifies 1 electric charge, unless it's something like calcium, which has 2 electrons that would give off. Again, I digress. Now, if we look at chlorine, chlorine just needs 1. It has 7 on the outer shell, so it needs 1. So its likelihood is that it would pick up an electron. So if it picks up an electron, would it be negative or positive? Right? So it's picking up an extra electron to fill that outer shell. If it picks up an extra electron, it would be negative. In this case, it would be called an anion. So chlorine, Cl, would have a negative to it. So given that, we know opposites attract, right? So positive and negative ions happen to bond with each other. And that's exactly what's happening in this case. It's called an ionic bond, where you get sodium that gives off an extra electron, and now it's positive. Chlorine picked up an electron. It doesn't have to be from that original sodium. It just picks it up from anywhere else. Picks up an electron, becomes a negative, an anion, and that cation and that anion bond together. And that's an ionic bond. And if you were to take a look at a bunch of sodium and chlorine atoms, they just happen to kind of attract each other and kind of cluster together. So this would be sodium chloride, which you might already know is table salt. It's exactly what it is. So that's ionic bond. On the other hand, we have something called covalent bonds. While ionic bonds resulted in the transfer of electrons somewhere where you had a positive charge and a negative charge, covalent bonds actually share the electrons. Okay, this is a different type of bond. And to give you an example, let's take a look at hydrogen atoms. Hydrogen atoms have on the outer shell only one electron. And as I mentioned earlier, the first shell needs 2 electrons. So in this case, that hydrogen atom, this hydrogen atom comes together, this hydrogen atom comes together, and what they end up doing is they share one shell. So at one point, the electrons over here, 2 electrons on this side, and then those electrons zip to the other side, and it satisfies both. So you get these electrons that are literally racing around their racetrack, being able to fulfill. So by sharing, they can fulfill each other, and therefore these bonds are formed just through that sharing of those electrons. In these cases, you'll see the term nonpolar, and I'll talk about that in just a little bit. Let me give you the example of oxygen. Oxygen is stable if it can share, if they can each share a pair of electrons. So here you'll see 4 electrons, it needs 2 on each side, so it ends up sharing 2 pairs of electrons. So you'll see those electrons forming back and forth, and what you see on here, so now that these oxygen atoms are now satisfied with the number of electrons on their outer shell, this forms a double bond. So this is a double covalent bond. Something we'll see with oxygen later on. Now let's bring it back to carbon. Taking a look at carbon, you'll see 1, 2, 3, 4 on the outer shell. So the fact that there's 4 on the outer shell, it would need how many electrons to satisfy the outer shell? It needs 4, right? So if it needs 4, it means it can form 4 bonds. And in most cases, that carbon will form a bond with hydrogen atoms. Those hydrogen atoms only need 1. What they can do is sort of share one of those bonds with that carbon. Another hydrogen can come and share, and 4 hydrogen atoms can come share together. So at this point right here, you can see our carbon atom. Carbon is now sharing electrons with 4 hydrogen atoms, and this becomes our methane molecule. Methane, such as methane gas. So carbon I'll talk about a little bit because carbon is an interesting atom for having 4. It allows us to build these huge molecules. Again, I don't want to spoil the fun on that, so I'll come to that in just a little bit. So I mentioned nonpolar because if we take a look at that racetrack that we just built around that hydrogen atom, those electrons spend equal time on both sides of the hydrogen atoms. They'll literally just race around the racetrack, and there's no point at which the electron spends more time around the hydrogen or the other hydrogen atom. That'll probably be better explained when we take a look at the next example of a polar covalent bond. And the best example for this is oxygen with 2 hydrogen atoms. So oxygen needs 2 electrons. Hydrogen has an electron that it can share. So in this case, oxygen binds to 2 hydrogen atoms. However, oxygen is a big atom. Same with phosphorus. And remember this because we'll be able to see this a little bit later on. Oxygen and phosphorus have a tendency to be electron hungry. They have a tendency to kind of pull electrons towards them. And if you look at the hydrogen atoms that form together, we create H2, 2 molecules of hydrogen, with 1 oxygen atom, H2O, otherwise known as a water molecule. And the sharing, the covalent bond that's sharing, we still see that those hydrogen atoms still share their electrons with that oxygen atom. But if you were to draw this racetrack, and you were to draw that electron that's moving around, which part do you think it would spend more time around? The hydrogen or the oxygen? Well, let me even draw something where it would really look like. Draw that racetrack. Now, where do you think it's spending more time around? It's spending more time around the oxygen. So if I have two electrons and they spend more time around the oxygen atom, that has a tendency to make the oxygen a little bit more electrically negative. Right? So if you spend more time around one side, it's going to make that electron spend more time around here and become more of a negative charge. And that term we call electronegativity. And that has a profound importance in the structure of the water molecule. In this case, the oxygen atom would be electronegative, which you can see the signal over here. And that's a sigma signal, electronegative. And conversely, because the electrons spend less time around the hydrogen, they have a tendency to be electropositive, which you can see again with the symbol above. So because the electrons spend more time around the oxygen, that produces a polar covalent bond, or a molecule with electronegativity, a side that happens to be a little bit more negative. And that, when you look at other water molecules surrounding, this is electronegative, and this is electropositive. If I were to take another water molecule, and this happens to be electronegative, and this is electropositive, again, opposites attract. Electronegative and electropositive creates a little bond. And that bond is hydrogen bonding. These are weak bonds that form between the electropositive hydrogen atom with an adjacent oxygen atom from an adjacent water molecule. And these are weaker bonds than the covalent bonds and the ionic bonds. In fact, they're fairly weak. But each oxygen atom can form 4 hydrogen bonds, 1, 2, 3, 4. So if you think of water molecules and they're constantly kind of pulling on each other, there's a lot of properties to water that you can see. And you can see that on our next slide. The first 2 properties I want to talk about cohesion and I want to talk about adhesion. And cohesion is water sticking together. You think of a couple and they might be cohesive. It's because they stick together well. On the other hand, you have adhesion. Adhesion is like an adhesive, like a tape that sticks to something. So in that case, tape would stick to something. In this case, what I'm talking about is water sticking to stuff. So water has that property of this stickiness, and that's related to the hydrogen bonding. So you'll see that since water sticks together, here you can see a water strider. This is a specific bug that walks on top of water. And the reason, if you take a look at the surface of the water, you can see that it kind of bends in a little bit. Those water molecules are holding together. Those hydrogen bonds happen to hold the water together, and the water strider can run across without breaking the surface of water. It's that light. It can do that. If you take a look at little dew droplets that form in the morning, that's the water vapor in the air that just happens to collect on surfaces. That's adhesion. That's water sticking to stuff. And then all of a sudden, the water droplets surrounding stick to one another and they start to become cohesively stuck together. Another example I want to use, if you ever wonder, I don't know if you guys ever wondered this, think of a plant and you water the base of the plant. Have you ever wondered, how does water get up the plant? How does it counter gravity to make it into the plant? Would you think you would probably want to pour it on the top to see if it somehow simmer down? Well, no, that's not the case. If you were to take a look at a stem of a plant, and here we have the roots, and here we have the water, and then right on top of the plant we'll draw one little single leaf. We have a bunch of water droplets that will start to stick to the inside of the stem. And what happens when you get sunlight that hits the top of the leaf, we know that's going to cause evaporation of water. Water starts to fly off. And as water flies off, what that ends up doing is it ends up pulling a whole string of water molecules up due to those cohesive properties. And it will pull from the roots and counter gravity. So that's a cohesion of water molecules pulling up that whole string of water beads all the way up. On top of that, if you remember from, if you ever measured things in a graduated cylinder, you probably ever took a look and you're trying to measure the amount of fluid, you saw that the edges kind of curve up against each other. That was a property of adhesion where water molecules stick to the surface of that cylinder. And here, if you remember, where were you supposed to measure? Well, it's due to the meniscus. That was what we called the meniscus. It was this curved area. That means that water happens to stick to the sides. So that plant happens to climb along. If you ever take a look at the straw, it happens to do the same thing. It literally will pull on the side, attracted to the stem walls, and attracted to one another as that water gets pulled out. So that's adhesion and cohesion. Other things about water. It is a universal solvent, meaning you take anything like salt, you take anything like sugar, you take anything like glucose, you take anything and you put it in water, and over time it will dissolve. It works really well for that. In fact, if you were to take a look at water, and I were to take NaCl, which was our salt, and we take water starts to come in and ends up surrounding that chlorine ion because of the electropositive and the negative. So the electropositive and negative have a tendency, and you'll see that hydrogen the water molecules surround that chlorine ion. So it'll break it apart. Water also has high thermostability. What do I mean by that? Water has a tendency to hold heat very well. If you ever think about why you sweat because you're generating heat. And the best way to get rid of that heat is a process of sweating. So that when you start to sweat on the surface of that skin, as water starts to pick up that energy that's being provided by that heat that you're generating, those water molecules start to jiggle around. And they start to break these hydrogen bonds, therefore releasing a good amount of heat. And as that water molecule comes off, you remove a certain amount of heat. Now that we're kind of like a little bit in a heat wave, do you guys remember ever going to like Magic Mountain or any like theme park and you step under those little mister things? Why does that cool you if they're just sprinkling just normal water on you? Well, when you stand underneath it, that water sticks to your skin, and eventually you heat it up and you evaporate. If you ever look at swamp coolers are I don't know if you still see them around anymore, but basically it's a big fan and it blows mist on you, and it should eventually just cool you off. Another thing to look at, if your engine ever overheats and you need radiator fluid, what's something you can use? You can use water. Water works so well to be able to hold heat. In fact, let's even take a look at temperatures. Has anybody been to the tropics anywhere? Like anywhere with high humidity? Well, how's the temperature over there? Always hot, right? At night, it happens to also be hot. It doesn't really cool down. And that's because the amount of water vapor in the air due to because of the high humidity, there's a bunch of water vapor in the air, and that water holds that heat well. So that when it turns night, the temperatures don't really fluctuate much. If you haven't been to the tropics, you've probably been to Vegas, right? You have less people there. Okay, more people that admit they've been to Vegas. In Vegas, you notice, especially in summer, it can be 110, 120 during the day. But at night, it drops down to like 80, 70. Right? So there's a big discrepancy between night and day temperatures. And that's because of the less water vapor in the air. So desert temperatures have a tendency to fluctuate quite a bit. Tropics, due to that humidity, have a tendency to stay. So that's the thermostability. We'll talk a little bit about reactivity and how it interacts with reactions taking place. And water also serves as a great lubricant. All right. So now that you have that, we've talked about the molecules. We've talked a little bit about building molecules. Let's start talking about chemical reactions, where we start taking these molecules and have them start reacting with each other, with the whole, and tying in the concept of what happens to that energy. Because this is going to have a profound influence on how when we eat, we take energy from it to use it to couple with the energy that we need to survive. So first, before I want to start out, I want to kind of talk about the key players involved. We have reactants and we have products. And any chemical reaction, I'm going to give you an easy example. A plus B forms C. And you'll see that the reactants are the starting substances, and products are the ending substances. So if I see A plus B, if I see A plus B, are these my reactants or are they my products? These are my reactants. And I know C will be my product. It's the ending substances. And in these reactions, there exists energy in the system that we can quantify. The first one we'll measure is potential energy. And potential energy is energy that's stored, and I use this term loosely, stored in the bonds, whether they're ionic bonds or covalent bonds. The real term for it, the potential energy, is the amount of energy stored as that state of being. Kinetic energy, on the other hand, is the movement of matter. So that's molecules moving around. They happen to kind of just jiggle around in their space. And the more they jiggle around, the more kinetic energy it has. So looking at that, if I were to take a look at, for the example of a roller coaster, and at the very top I have that roller coaster cart that's just sitting right at the very top. It's not moving at all. It has a high potential energy, in that that's the energy that it could use to start to fall down. So if I were to take my remote here, it has a bunch of potential energy. It's not moving, so its kinetic energy is 0. But if I let it go, and it's about to hit the ground, that potential energy is decreasing, while the kinetic energy, the energy of movement, is increasing. So in that case, you'll see what I was talking about earlier. High potential energy, high potential energy, low kinetic energy. And when I drop it, when that roller coaster gets down to the very bottom, let's just say low potential energy, high kinetic energy. The energy transfer that happens back and forth is conserved. And that is our first law of thermodynamics. Whenever we look at a system, when we move from the reactants to the products, the energy is always conserved between the two. There's no loss of energy, and there's no gain of energy through this reaction. It's always going to be transformed from one to another. But if you're to use that example and you think about that roller coaster, if that were absolutely true and that were the only law of thermodynamics, that would mean that this thing could go on forever and it would non-stop move over and over again. But there's a little bit of loss to this energy. And I want to use, again, that term loosely, of loss of energy. What's given off over time? If you were to touch the wheels of that roller coaster cart, it would be hot. There's a certain amount of heat that's given off. And that's part of the energy transfer that occurs. There's a loss in potential energy that happens due to heat loss. If you ever drive your car for a long distance, you know your car gives off a certain amount of heat. But if you were to measure that energy that moves from this point to this point, and also add the amount of heat loss, that energy from this point to this point, potential energy, kinetic energy, plus the heat that's given off, the potential energy in this case, plus the kinetic energy, plus the heat, those two energies, the sum of both energies, would be the same. So that's the second law of thermodynamics. Another idea is to take this again into something physiology related. If you were to think about the food you just ate, right? However long ago. You take the energy from the bonds that are in that food and you break it apart, and then you use that energy. You don't actually use all of that energy that you take from food. Some of that is actually given off as heat. If you don't believe me, feel your own skin. You're giving off heat. That heat, in fact, we're not even really all that efficient in terms of converting all of that food to potential energy that we can use for growth, reproduction, movement, building of proteins, building of stuff, repairing ourselves, etc. We actually give off a good amount of heat. In fact, if the amount of food that you eat, 25% actually gets stored as potential energy. The other 75% we actually give off as heat. And we do that for two purposes. Number one, to keep your, well actually I only know one right now, is just to keep your body temperature at about 98.6 degrees. So that's the second law of thermodynamics, is the decrease in potential energy. If you go into the book, it talks about entropy, and that's entropy is a measurement of the measure of disorder. I don't want you guys to get too lost on that concept, but basically when heat disperses, it will travel. Like if I give off a certain amount of heat right here, or if I were to have like a cup of boiling water that's giving off heat, eventually that heat will continually disperse. That's the disorder that comes from it, and it will disperse into this room until it's all equal. The third law isn't even really physiologically relevant, but it's basically us saying we can't get to the coldest temperature ever, which is like absolute zero. That comes to a certain point. It's a theoretical point where these molecules have zero kinetic energy. And the molecules, they normally jiggle around, and eventually you get cold enough that the molecule just stays still. And that we can't really get to. We've had laboratory settings where we try and get as cold as possible, and it's really, really close. We just can't get to that point. It's almost like, I guess in ways, kind of like stopping time. But again, I don't want to digress too much. Okay, so now I've talked about this as a recap. A plus B are reactants, and C is our product. And I want to use another example to help really tie this in. And that's going to help us explain some of the other concepts that we're going to be looking at. Because what we're doing is we're going to be building on top of this. A plus B, we're just going to use the example, and I'm actually going to just draw this on the board. We'll do A plus B, where A equals man and B equals woman. And then these are our reactants. And then C equals baby. Okay? And this will be our product. So again, I'm just using this as an example. But in this case, A plus B becomes our product. So in this example, you just have to pretend that man plus woman form together and they produce baby, and then the man and woman are expended in them. They just disappear into the thin air. They get destroyed in the process. So man plus woman becomes baby. So think about that as we move on to the next slide. So in this case, we're going to take a look at the amount of potential energy in a reaction. What's needed in order to go from man plus woman, which are reactants, to turn into our final product called baby. And there's a certain amount of energy that's going to take place. There's an energy transformation, where if we measure the amount of energy here, plus the energy here, they're going to be equal to one another. So in order for this reaction to take place, we have to measure the amount of potential energy. Just like we saw on the prior slide, in order to get from here to here, we want to measure the potential energy at section A, and we want to measure the potential in section C. That gives us the sort of progress on this reaction taking place. So what I want to look at is the measurement of potential energy for A plus B, and then our final energy in our products with C. So man plus baby, here's the energy of the reactants, and then C is the energy of the products. And you're probably wondering, well, didn't you just say that the energy should be equal? Well, that's true. But there's a loss in potential energy, which means there must be a gain of some form of kinetic energy. And that energy could be heat in this case. So this, from here to here, this is the level of the products, and this is the energy level of the reactants. This right here is a negative value. This is the amount of energy that's lost. And you don't really have to know this term, delta G, but that's the change in potential energy. And we have a negative change in potential energy. So by measuring the potential energy, we would see that typically in this case, and let's use that example earlier at some point. A plus B, you have a bunch of A's and B's in a cup. Let's just put a bunch of men and women in a beaker, or a room, or whatever you want to call it. A bunch of A plus B, A, B, A, B. And think of these men and women just going around randomly, just moving around like this. They're just floating along, and eventually, they might just get in the right position and bump into each other and start a reaction. And that reaction, when A and B come together, they form what again? They form C, they form baby. So now we're going to wipe that out, and we're going to form now C. Okay. So the amount of energy that's required, there's a certain amount of energy that you need for those two to get together. They don't just bump into each other and just form baby all of a sudden. They have to get into the right configuration, the right place, and eventually there's a certain amount of energy that's required to start that reaction. If that doesn't make sense, think of a fire. I have a bunch of paper on my desk right now, and I want it to catch on fire. It's not going to just spontaneously catch on fire. There may be an off chance that somehow some ray of light comes in and might spark something up, but really what's needed is some form of energy to get the reaction to start. It's just a little bit of energy that you need to invest to get it started. So in this case, I might have to use a match just to get it to light up on fire. Or if I were to take that roller coaster, for instance, that roller coaster I mentioned earlier, that thing might just be sitting there. And in order for it to get down to this point, you gotta wait some time. Eventually, maybe if the conditions are right, maybe somehow it just starts and then it starts to fall. The activation energy is that little push that's required just to get that energy started. That means you need to put in a little bit of energy in order for those things to occur. So in this case, these products, A and B, or I'm sorry, the reactants in this case, have to have a little bit of energy to get into the mood to eventually be able to form our product. Now think about things that would speed up the process. And what I mean by speeding up the process is the speed at which this reaction takes place. A plus B turns to C. If I start increasing the number of our reactants, putting a bunch of men and women into this beaker, there is a higher likelihood that our products start to form at a faster rate. Why? Because there's a bunch of them. There's a likelihood that they'll just bump into the right configuration, and then these form a reactant, or a product, and then these form a product, and then these form a product. So that speeds up the reaction. You want something to happen a little bit faster, add more reactants to it. Something's not burning fast enough, the paper's not burning fast enough, add more paper to it. That's one way. The other way is to increase the temperature. If I increase the temperature, these products start moving around faster, and they move around faster, and they move around faster, and eventually there's going to be enough energy that they bump into each other fast enough that they create products faster. If that concept doesn't make sense, think about what happens when you're trying to dissolve a bunch of sugar in water. You think this is taking a long time. What happens if you heat that water up? You start introducing heat, those water molecules start moving around, and then whatever your sugar is starts to get dissolved faster, because those molecules bounce around faster. So increases in concentration and then increases in temperature will speed the reaction further. And then to even take it a little bit further, let's say if I were trying to slow down the concentration, or slow down the reaction, and I've got a bunch of, it's been some amount of time, and eventually I've got a bunch of product around. Do you think this is going to speed up the reaction or slow the reaction down? Let's slow it down. Because A can't bond with C, B can't bond with C. These are our products. It's only man plus woman that will start to form baby. We can't do man plus baby. That doesn't do it. So, in this case, the rate of product formation, when we start to have a bunch of products in there, that slows the reaction down. That, if you go into the book, is le chateleurs, and I can't pronounce that right, or even spell it right, but you get the general gist of it. The principle in that A plus B forms C, you increase the amount of C, and that slows the reaction down. You get less A and B. You want to speed up the reaction, get rid of the babies in there, and eventually you'll get more reactants that bump into each other. What questions do we have about this concept before I continue? No questions? All right. So remember this slide, because I'm going to pull up this in just a little bit. We're going to add on to this concept. So now that we've talked about energy transfer, let's look into ways energy can be transferred in or out of our reactions. And if you're to take a look at the two types of reactions we have endergonic reactions and we have exergonic reactions. And the main difference between the two is the direction of which energy is going into the reaction or exiting the reaction. Endergonic reactions require energy to be put into the reaction in order for that reaction to take place. Exergonic reactions are reactions in which one of the products is the release of energy. So looking at that example I talked about earlier, A plus B plus a certain amount of energy produces baby. This is baby turning into man plus woman and releasing a good amount of energy. To kind of give you an idea, this is how fuel works. This is how dynamite works. There's a good amount of energy in there, and once you start to break them apart, you release a good amount of energy. And that energy that's being released is released from the bonds that are formed within the baby, typically where that stuff happens. To give you another example, this is that hamburger. Now I'm just thinking hamburgers, I had In-N-Out last night. That hamburger being broken down and releasing amount of energy from it. I'm breaking that hamburger down into its smaller constituents and releasing energy. This endergonic reactions is where you have energy being put into the system. So before I go a little bit further, let me help you clarify these two terms, endergonic and exergonic. How can I remember the difference between endergonic reactions and exergonic reactions? I highlighted ender and exer for a reason. Ender for entering energy. Exer for exiting energy in the reaction. That's another mnemonic that I'm giving you. I think this is number 3 now. And in endergonic reactions, you'll see these are typically referred to as anabolic reactions. We call it anabolic, that's a term anabolic, like anabolic steroids to build. It's exactly what we're doing. We're taking small components, A plus B, and forming it into one bigger molecule. So this is an anabolic reaction. And in this case, in order to get an anabolic reaction to take place, you got to invest some energy into it. It's an endergonic reaction. And in the same token, from looking at from an energy perspective, you also see the term dehydration synthesis. Dehydration synthesis. This is the synthesis of molecules that requires dehydration. So in this process, do you think you're losing a water molecule or you're gaining a water molecule? You're losing a water molecule. So this is loss of water. Again, not wholly important, but I will show you in some of the reactions, I'll show you where there's a loss of water molecule to put these two together. Now, exergonic reactions are catabolic reactions. This is where you're breaking apart those bonds. Yes?
Can you, where this is synthesize a large molecule from a simpler, smaller molecule? Like, I don't understand that. Like, making simpler or...
Putting a water molecule together, or putting molecules together.
So basically, you're starting from creating a bigger molecule by putting smaller molecules together.
Yeah, like think of like muscle, for instance, putting proteins together. Think of building building a starch molecule. You're putting things together. You're constructing them. You're taking, and we'll be able to see these molecules a little bit later on. So hopefully that'll clarify that in just a bit. Now, the exergonic reactions is the release of energy. So in this case, we call them catabolic. So this is breaking them apart. And in this process of breaking apart those bonds in a molecule, you're releasing energy. And that release of energy can be used to be coupled with another type of reaction. What do you think that would be? Well, if this is releasing energy, this might be requiring energy. So that same energy where you're breaking apart energy allows you to create endergonic reactions that require energy. So think of what you're doing when you're trying, let's say you have cells that are growing, replicating. They require energy. So the breaking apart of energy, if you're going to take sugar molecules or molecules from the food that you just ate, you're breaking that apart to couple that with reactions that require building. So the combination of endergonic and exergonic reactions are exactly what metabolism is. And we're going to learn more about this in next Thursday's lecture, when we talk about cellular respiration, which is the process of breaking apart a glucose molecule. We're breaking apart a glucose molecule into its smaller products to release a good amount of energy. And that energy, which we'll talk about next Thursday, is a molecule which we'll call ATP. Again, we'll talk about in a little bit in today's lecture. It's a form of energy. So in this case, you'll see this is in exergonic reactions because it releases energy. It's called hydrolysis. The term lyse, again, something I'd like you guys to be familiar with. It's something you're going to be able to see later on. Lysing is to break something. And it requires hydro. What do I mean by that? Water. Water lyse. A water molecule is introduced into a molecule along to start the reaction to break it apart into its smaller constituents and release energy. So now that we've talked about the reactions, the reactions are where we have endergonic and exergonic reactions. Let's take a look at molecules. We'll take a little jump and take a look at the molecules that can be formed. And again, we can break these down into inorganic compounds and organic compounds. Inorganic compounds are molecules that lack a carbon backbone. This will make much more sense when we look at our carbon-based organic molecules in just a bit. But inorganic compounds lack that carbon backbone. It doesn't mean they don't have carbon, it just means it doesn't have this carbon structure. So with that, we can see what types of molecules we have. We have water, H2O. We have acids, bases, and salts. And vitamins and minerals typically are typically inorganic. So here you can see with acid, bases, and salts, something I won't really get too much into, because again, this is something that you saw from chemistry. We can measure acids and bases. Acids in a process that releases hydrogen ions. This is an example of an acid. Base, which contains a hydroxide anion that has a negative charge. And we can measure the pH of a sample by measuring the concentration of the hydrogen ions. Think of sulfuric acid, you think of carbonic acid, you think of just battery acid, you think of any type of acids, and they give off these hydrogen ions. And we can measure that. And in fact, pH is on a negative scale, meaning the lower or the higher the acidity, the lower the pH. It actually exhibits a logarithmic scale, which you don't really need to know, but pH, neutral pH would be a pH of 7. That means this is fairly neutral. We know that things that have a higher pH are more basic. So anything about 8 plus is going to be basic. And anything below 7, anything less than 7, is acidic. So we can measure that based on that pH. And the importance behind this, the importance in our physiology, is just like we talked about on Tuesday. We have a narrow window that we typically will consider normal. And blood has a pH of 7.35 to 7.45. If you don't believe me, take a look at your labs when they're drawn, and they will measure the pH of your blood. Anything below 7.35, remember, negative logarithmic scale, anything below 7.35 is acidosis. Anything above is referred to alkalosis. So, low pH, acidosis. Higher pH is alkalosis. And if you were to measure somebody's pH of their blood and it drops, that's a condition called acidosis. And they typically want to find out what's contributing a bunch of hydrogen ions into the bloodstream that's causing it to be slightly acidic. And there's metabolic acidosis, there's respiratory alkalosis, there's a bunch of causes for the disruption in the pH. And it causes a whole host of changes that happen physiologically, which we won't really get into right now. But we do have a buffer in our blood that prevents the changes. You would think, well, if that means if I just add a little bit of acid to my blood, is that going to change my pH? Does that mean I'm going to die? No. We have buffers in our blood that prevent minute changes in the pH. And that buffer is bicarbonate. This example right here, H plus HCO3 becomes carbonic acid, is something if you don't get now, that's okay. Because we're going to beat this whole concept down over and over and over again. This is the fundamental concept in physiology. It's carbonic acid. When you get CO2 plus water, it forms a weak acid known as carbonic acid. And that carbonic acid in water can dissociate into two ions, a hydrogen ion and a bicarbonate ion. That bicarbonate ion can attach to other hydrogen ions. So if you were to inject a little bit of acid into somebody's blood, those bicarbonate ions would attach to it to neutralize that acidity. And then, what happens with that other hydrogen ions? Well, we can get rid of this in multiple ways. You'll learn in our respiratory lecture, we can get rid of hydrogen ions this way. You'll learn in our urinary lecture, we can get rid of hydrogen ions through urine. You'll learn through the digestive system, we can get rid of hydrogen ions there as well. We have multiple ways of getting rid of it. That's the whole focus of what we're going to be talking about in our urinary lecture, is the getting rid of acid. But what's left over is this bicarbonate ion. That's that buffer that prevents any changes to keep us within that narrow window of 7.35 to 7.45. So that is our example, our first example of homeostasis in blood. And again, we'll go over this in our cardiovascular lecture when we talk about blood. Okay, now on to organic compounds. Organic compounds are compounds that contain a carbon backbone. And what do I mean by carbon backbone? Remember what we talked about earlier about carbon, where it contains 4 electrons on that second outer shell. It can bind to 4 other bonds, 4 covalent bonds with another atom. And in most cases, you'll see that it forms with carbon molecules. It's our way of, if you were to take an example of aliens, if aliens existed and they came down and they sampled humans and they found out what causes, not even just humans, but all life form on this Earth. We know that the Earth is comprised of 70% water. But what makes us really unique is that we are carbon-based forms. We have carbon atoms that attach to one another to create these huge molecules. Okay? It's one thing when they're looking at Mars and they're trying to find water, and they say, okay, water, great. Water is something because it has those properties that we just talked about. But then the next question, are there organic compounds that exist? Because that means if there are huge carbon molecules, that means there had to be some chemical reactions that took place to create those carbon atoms, to create these huge organic compounds. And if something had to create it, that means that something could be sped up with stuff that's produced, possibly by life. Again, I don't want to digress too much. I want to focus specifically on our organic compounds. But carbon can bind, let's say, to another carbon atom, and that forms a covalent bond, C and C. And that's one covalent bond that forms. And now, one thing I want to point out is now we have 3 electrons from each carbon that can bind together. Typically, what would happen is hydrogen can bond to each of those free bonds, or free electrons. And I'll talk about that in our next slide. That's our alkanes. You'll see that the formation of these huge carbon atoms, and in fact, that C can bind to C, can bind to C, can bind to C, can bind to C, and you get a bunch of H's wrapped around. You guys get the idea. Another way of drawing it, if you remember from chemistry, is that you can draw these signs. You guys remember these? And each of these points right here are carbon atoms. So this serves as the foundation for huge molecules that we can create. And the 4 main organic compounds that we'll be looking at are carbohydrates, proteins, lipids, and DNA. So this is kind of funny. Whenever you go to the grocery store and you see the term organic, typically if you were to see organic, you kind of want a definition of what they mean by organic, because this is a term you've probably seen abused quite a bit. Just because a fruit is organic, yeah, of course it's organic. It contains organic compounds in it. There's a bunch of sugar molecules in it. That makes it organic. But that's not the term they might be referring to. Organic referring to as no pesticides being used or whatever other organic things are terms are being used. That's the whole concept behind organic. It's just carbon backbone molecules contained within. So with that, now I want to take a look at that molecule that I pointed out earlier, our carbon that's bound to a bunch of hydrogens. This is what we call alkanes. And this is what's called hydrocarbon. They're called hydrocarbons because we have a bunch of hydrogen and we have a bunch of carbons. And it's exactly that molecule that I just drew over here. I have 2 carbon atoms bound together by 1 covalent bond, bound to a bunch of hydrogens surrounding. And in this case, if you were to take a look at 2 carbons and 6 hydrogens, 2 carbons, 6 hydrogens, you have what's called ethane. If you get 3 carbons and 8 hydrogens, you have propane. You have methane, butane, pentane, hexane, octane. These are all your fuels that you would have heard of. These things burn off and release a good amount of energy. You've probably heard of octane. This is stuff in gasoline when they're measuring. That's our hydrocarbon. Now we're going to take that same structure that I talked about earlier, and instead of hydrogens on one of the connection points, I'm going to leave this one free. There's a weird thing on my computer that it just keeps erasing. Anyway, there's this point right here that we're going to just talk about right here. And I mentioned this on this last slide right here. Functional groups. Functional groups is a way of taking a molecule, taking one of those slots that attaches to carbon, and attaching a group on there that would change the characteristic of that molecule. Of which we have 1, 2, 3, 4, 5 main ones that we're going to be talking about. These are things that can attach to that point right there. And one thing I'm going to talk about is one of the functional groups is OH, a hydroxyl group. So instead of a hydrogen, I'm going to put OH on there. And in that functional group, we have OH. That becomes our alcohols. So if I have ethane, and I attach a hydroxyl group, which makes it an alcohol, this would be called ethanol. Has anybody heard of ethanol before? Ethyl alcohol. That's the stuff in alcohol. Right? That's ethanol. And that changes the characteristic. Where this was a pure fuel, this becomes an alcohol, which also serves as a fuel too. The other thing that we have carbonyl groups, and you'll see some carbons that have a double bond to an oxygen. Another one is a carboxyl group, and that is a carbon double bound to an oxygen bound to a hydroxyl group. So these are typically called acids, a carboxylic acid. Typically they would be acids because that hydrogen can break off and create a hydrogen ion. But another way of remembering carboxyl group is to, let me just erase this H real quick, draw it up here, and this creates a little box, carboxyl group. Amino group is another functional thing that we're going to be able to see. Take off that hydroxyl group and add an amino group. And this is going to serve as a foundation of what we're going to be talking about later on. So just remember this molecule for now when we talk about this towards the end of our lecture. This is going to serve as the foundation of protein. And this creates an amino acid. The last group is sulfhydryl, SH. And sulfhydryl groups contain sulfur. So typically compounds that contain sulfur on it have that smell. Do you guys remember those, I don't know if you guys ever played with these, they were like the stink bombs? Yeah, and you drop them, and then you get this concentration of sulfhydryl molecules in there that start to disperse, and once you pick up on it, you smell the smell of rotting eggs. Sulfur is that exact smell, and that's a way you can remember a sulfhydryl group is SH. If you just write IT at the end, you'll remember what it smells like. That's exactly it. Okay? When we get into our, specifically our sensory lecture, we'll take a look at the molecular compounds that are found in things that smell. Body odor. You'll see tons of sulfhydryl groups on there. But sulfur doesn't just form the process of smelling like things. In fact, something that you will see in especially molecular biology is that it forms very strong bonds called disulfide bridges. Something that you will really see, especially in our protein section that we'll talk about later on. You get one sulfhydryl group and another sulfhydryl group. They can break those apart and create these strong bridges that will hold a molecule together. So with that, it's been about an hour and a half. Let's take a break before we get into the last half of today's lecture. Should we take a 10-minute break? Is that good enough time? 10? 10? Yeah? 5? 10? 10? 5? 10? 10? Okay, 10 it is.